name is Jonathan Adorno. I go to Northeastern University, and I currently do research at Lawrence Livermore National Lab, where we're trying to develop a tumor on a chip, which is going to be an ex-vivo tumor model system for breast cancer. So initially, we were trying to figure out how are we actually going to bioprint this tumor. So we have to figure out what's going to be the bioink, how are we going to what are we going to put these into. So we would initially culture the spheroids and then collect them and then place them into these hydrogels of collagen and fibrin. And if you look here, you can see that in collagen, the same spheroids versus in fibrin are invading, versus in fibrin, which they are hollowing out and growing like a balloon. Which is interesting because it's the same spheroid, just different materials they're being placed in. Versus when we put them in a bioreactor where culture conditions are different, you can see that farther from the channel, which is right here, if they're farther away, they actually grow towards the channel, which is more of the invasive phenotype we saw with the collagen, versus if they're closer to the channel, they just balloon out again. And our gel is supposed to be opaque, but you see that these are actually clearing out. So the spheroid's also interacting with its environment, which you can see here, which we tested to see if they're laying down any basement membrane protein, in this case laminin, and we saw they actually are, and they're still growing. And so with all this information, I was able to put it all together and then develop a bioink. And so in the bioink, after playing around with concentrations, I was able to get something that I could extrude and have the cells still alive in. So if you see here, we have these lines that I was able to print. Since then, we've played around with the concentrations and made them even better lines. But it'll help us to place the spheroids in specific locations so that we can always have more replicative data and eventually integrate these sensors that we're trying to put in at Lawrence Livermore National Laboratory that we make on site so we can get real pH and oxygen readings. And everything we do, we compare back to our mice models, and everything has been very similar, and we're very excited to keep this going.
Thank you very much.
Yeah, no problem. Thank you.